Hi, Hannah. Hi, everybody. We'll just give everybody a minute to join in as usual. I hope everybody's doing okay. I hope you're managing all right. I know people are starting to get frustrated and bored and cooped up, and it's not an easy thing we're all going through. But hopefully we can get a little distraction with schoolwork just to take our mind off it for a little while. Let's see if I can get a little more light here. Okay. I agree. I agree. It's been very nice to have work to focus on right now. You know, I have a good friend, Hannah, who I saw yesterday. We took a walk together, you know, 6 feet apart from each other. We were walking, and she told me she's really enjoying the quiet, the isolation that she's having right now. Because she normally, I guess, has a very busy, loud, you know, constantly going life. And she said she's just kind of enjoying having a little quiet right now. All right, we're going to go ahead and get started. And then, as you know, people can join in as they arrive to the meeting, as usual. Today, we're going to be talking about protein and protein catabolism. If you look in your lab manual for today, there's actually 2 exercises for protein catabolism, part 1 and part 2. And on our original schedule, we were going to do those 2 exercises on 2 different days. But since our schedule got a little compressed, we're going to do protein catabolism today as 1 exercise. So it'll be parts 1 and 2, but we'll do it a little bit differently so we have enough time. One of the things that we're going to do is we're not going to look at every test that is discussed in lab 1 and lab 2 for protein. We're only going to talk about the ones that are a little more common. And the other thing we'll do is we'll talk about the way that we interpret these tests, but this time I'm not going to give you results to enter into your lab manual. We're just going to look at how to interpret them, what they're testing for, and then I'll tell you what questions that you'll need to complete in your lab manual. So to get us started, So we've been talking about metabolism in terms of carbohydrate. And we've talked about how different microbes use carbohydrate as a source of fuel, as a source of energy. We talked about all the different tests involved in identifying whether or not a microbe is capable of using certain sugars and whether they're using them for respiration or for fermentation. So today we're going to move over and talk about protein. Now, we need to nail down a couple of definitions about protein because you hear these words and you'll hear me use these words, and they do actually mean different things. So this word peptide refers to a short chain of amino acids. And by short, I mean somewhere in the neighborhood of tens to hundreds of amino acids. You can call that a peptide. So when a textbook talks about a catabolic reaction, creating small peptides from protein, what they mean is the protein is being broken down into smaller chains of amino acids. The term polypeptide is essentially a long chain of amino acids. And by long, again, we're saying somewhere in the hundreds of amino acids to the thousands of amino acids. But when you hear the word polypeptide, they're specifically talking about the chain of amino acids that is produced during translation. The term protein technically means a folded up, functioning polypeptide. So a polypeptide isn't functional until the cell has folded it all up into its three-dimensional form. Now, I am very guilty of using the word protein when I should be using the word polypeptide. You'll hear me use those two words interchangeably, and I really shouldn't, because a polypeptide is not yet functional. It has not yet been folded up and processed in a cell. So sorry, go ahead.
Really quick. So you're saying that a protein is just a folded up, ready to rock and roll polypeptide.
That's exactly right.
Okay, thank you.
A polypeptide is what is actually produced during translation. The ribosome is making a long chain of amino acids. But then the cell, in order to make use of that, the cell has to fold it because it doesn't become functional until it's folded up. Good question. So if you look at part 1 in the lab manual, and that's starting on page 125, the first exercise in protein catabolism is about the ways that microbes catabolize protein. And primarily, we're talking about microbes secreting enzymes into the environment so that they can break down these big, bulky proteins into either smaller peptides or into individual amino acids. And then the cells can take those peptides or those amino acids in and use them for metabolism. So remember, when a microbe excretes or secretes an enzyme so that it can work out in the environment, we call that an exoenzyme. An endoenzyme is an enzyme that functions inside the cell itself. So this is about catabolizing protein out in the world, basically, so that the microbe can make use of that protein. Proteins are generally enormous molecules compared to little microbial cells. So it would be really hard for a microbe to take a big intact protein across its membrane. So instead, it breaks it down out in the world and then brings in the pieces of the protein, the peptides or the individual amino acids. Now we're going to look at two tests with these exoenzymes. We're going to look at what's called the gelatin hydrolysis test and the urease test. So what the gelatin hydrolysis test involves is growing some bacteria on an agar plate that has gelatin in it, and then looking to see, as the cells grow into colonies, looking to see if the gelatin gets broken down. Now, if you're not familiar with gelatin, gelatin is what we would call a lower quality protein. It's animal sourced for the most part, and it comes from parts of the body that we don't tend to eat as meat. I'm trying to be as diplomatic here as I can, but gelatin is kind of a nasty sort of a waste product from animals. And yes, that's what goes into Jell-O and marshmallows and other things. It's just a low quality protein. When I talk about a protein as being low quality, what I mean is it doesn't have a very wide array of amino acids in it. A high quality protein, and that would be something like, let's say, cow's milk or egg. A high quality protein has a vast array of amino acids in it. And if you think about it, cow's milk is intended to nourish a calf. It's intended to take care of a calf nutritionally in the first weeks of life. So it has to be a nice, complete, high quality protein. It has to offer a whole range of amino acids. Gelatin's very different. Gelatin is sort of a very basic, low quality protein, but it's still a protein, and microbes can use it as nutrients. So there's an enzyme that some microbes have called gelatinase. That's a great name for this enzyme because it tells us exactly what it does. Gelatinase breaks gelatin down into individual amino acids. And then the microbial cell can absorb those amino acids and use them. Now, the urease test is very different. Urea is a molecule that is created from the metabolism of protein. So when a cell, for example, a human cell, breaks down a protein that has been consumed, one of the byproducts of digesting a protein, if you will, is urea. And urea, again, is a waste product. So there is an enzyme called urease. It's another exoenzyme for our microbes that is capable of breaking apart urea and creating ammonia. When you change a baby's diaper, I know some of you have kids at home, when you change a baby's diaper and it's wet with urine, you know that ammonia smell that hits you when you get ready to change the diaper. There are bacteria in the baby's bladder that are breaking down the urea in the urine with urease and creating ammonia. And that's the smell that we associate with urine. So two tests that are looking at catabolism. One of them is to see whether or not the microbe can break down gelatin. And one of them is to see whether the microbe can break down this waste product called urea. And I will mention that while you and I might think of ammonia as being just this nasty, smelly waste material, to a microbe, ammonia is a nitrogen source. So to a microbe, ammonia would be a useful molecule. So it makes sense that a microbe would want to break down urea. All right. Now, in the second exercise in your lab manual, instead of talking about catabolizing protein, they discuss catabolizing individual amino acids. It's important for us to remember here that the most common use of an amino acid in a microbial cell is to build new protein with it, okay? Most of the time when a bacterial cell absorbs amino acids, what it's going to do with those amino acids is use them for an anabolic reaction. It's going to use them to build its own protein. But it is possible for a microbe to take an amino acid and break it apart. Basically, take it apart and use it for carbon and for energy. So it's a much less common fate of an amino acid to be broken down like that. But our microbes do it, and we do it as well under certain circumstances. So the second exercise in protein catabolism is in the catabolism of amino acids. Now, cells break down amino acids in a couple of different ways. And your lab manual goes into these details as well. There's a process called deamination. There's a process called decarboxylation. And there's a process called desulfuration. And these are all ways that a cell could break down an amino acid. So deamination, like the name suggests, removes the amino group from the amino acid. And we'll look at the structure of an amino acid in just a sec. So it's going to take off that amino group from the amino acid. And that's going to create two things. It's going to create an acid and ammonia. Now, we don't need to learn any names of enzymes for deamination because it turns out that there are different enzymes for each amino acid that gets deaminated. So we're not going to learn those names in any way. But I do want you to be familiar with the name of the process and the products that are made, because those are going to be used in tests to see whether or not deamination is occurring. Now, decarboxylation, as the name suggests, is a process that removes the carboxyl group from an amino acid. And this time, the product that will be produced is a base, what we call an amine, and carbon dioxide. And again, a whole range of enzymes do decarboxylation, a different enzyme for each amino acid, essentially. Now, the third process for breaking down amino acids only applies to a couple of amino acids. Remember, there are 20 of them altogether. This one only applies to amino acids that contain sulfur, and hence the name desulfuration. For example, cysteine has sulfur in it, methionine has sulfur in it, tryptophan, I believe, has sulfur in it. So a handful of our amino acids contain sulfur, and some of our microbes can go through this process of desulfuration to break those amino acids down. Now, when that happens, the amino acid is essentially broken down to produce a very noxious smelling chemical called hydrogen sulfide, or H2S. There is a specific enzyme that does this reaction. It's called desulfhydrase, and I have the name here on the slide. Desulfhydrase can break down a sulfur-containing amino acid and produce this very nasty smelling hydrogen sulfide. I should mention that when amino acids get broken down through deamination or decarboxylation, the acids and the bases that get produced tend to really smell bad. You'll notice in your lab manual, some of these acids and bases have these hysterically funny names. Like there's one called putrescine. There's one called cadaverine because it smells like a cadaver. So these metabolic products of the breakdown of amino acids tend to be very smelly, not a pleasant thing to smell. All right, let's review what an amino acid looks like. Remember, all amino acids have the same general structure. There are 20 different amino acids, but they all have the same basic structure. They have a carbon right in the middle, and then attached to that carbon, there is a single hydrogen, and then what we call an amino group. That's the NH2. And then what we call a carboxyl group. That's COOH. And then the last thing that's attached to the carbon is the R group, or the functional group. If you recall, when we were talking about biochemistry, we said that functional groups are the parts of molecules that give the molecule its own unique chemistry. And it's the functional group that determines whether or not an amino acid is polar or nonpolar or acidic or basic. It's all about the R group. Now, when microbes break down an amino acid, remember we said sometimes they use a pathway called deamination. That will remove this end of the molecule and create an acid. Decarboxylation will remove this end of the molecule and create a base. And then desulfuration is only done on sulfur-containing amino acids, and the sulfur is found in the R group. So desulfuration hits the R group. All right. So for the second part of the protein catabolism exercise, we're also going to look at two tests. We're going to look at a test called phenylalanine deamination. And this is a test that, as the name suggests, tests whether or not a microbe can deaminate this amino acid called phenylalanine. Remember, deamination produces an acid and ammonia. The acid is called phenylpyruvic acid. And then that phenylpyruvic acid, we can't see in the tube. So in order to determine whether or not it's there, when we take the tube out of the incubator, we're going to add a reagent to the tube. We're going to add in what's called ferric chloride. It's an iron-containing chemical. When you put ferric chloride in where phenylpyruvic acid is, the two things are going to react with each other, and they're going to create a dark green color inside the tube. And of course, we can see that. So if we put the ferric chloride in and we see a dark green color, we know that our microbe has performed phenylalanine deamination. The other test is a test for hydrogen sulfide production. And specifically, it's looking for the desulfuration of the amino acid called cysteine. Now, there'll be an acid produced here as well, and that one is called pyruvic acid. And remember, pyruvic acid is just what we've been calling pyruvate. So this is the same molecule that we talk about when we talk about cellular respiration, pyruvate. So during hydrogen sulfide production, during the desulfuration of cysteine, some pyruvate will be made, and some hydrogen sulfide will be made. Now, once again, we can't see the hydrogen sulfide. So when that tube comes out of the incubator, we add a different kind of iron compound. And this one is called ferrous sulfate. We add in some ferrous sulfate, and we look for a black precipitate inside the tube. The tube really does, it turns black with this reaction. So 2 reactions where we're going to look for a color change. All right. So let's take a look at how we interpret the tests that we've talked about so far. Remember, the first test for protein catabolism is this gelatin hydrolysis test. Now, when you take this gelatin, it's usually in a tube, a test tube, and that's what you're looking at here in the picture in B. Gelatin is a little bit like agar, right? I mean, it may have been a while since you made some Jell-O at home, but it has a very similar consistency to Jell-O. I mean, to agar. Here's the big difference, though. The reason that we use agar to grow bacteria on and not gelatin is because agar doesn't get degraded by bacteria, right? And remember, agar is going to stay solid even when you put it in an incubator. The same is not true for gelatin. Gelatin will melt when you warm it up. Gelatin will become a liquid when you warm it up, and some microbes can degrade it. So here's how it works. When you take a gelatin hydrolysis tube out in the lab, before you inoculate it, at room temperature, in other words, the gelatin is going to be solid, just like you can see in tube B. Then we're going to inoculate it with some bacteria. We're going to put it to incubate, and then we're going to check to see what happens to the state of the gelatin. We expect it to be solid at room temperature. We expect it to melt in an incubator. So what we do is we take the inoculated tube and we don't put it in the incubator. We let it incubate at room temp. We put it over on a corner desk and we let it incubate. Now it's going to take longer to grow bacteria at room temp. And in fact, we usually leave these gelatin tubes out to incubate for about 7 days, for about a week. Now, if the microbe creates this and secretes this enzyme called gelatinase, the gelatin is going to turn to liquid in the tube because the enzyme is breaking apart the protein into smaller pieces. It's breaking it apart into individual amino acids. And then, of course, the microbe can absorb those amino acids. So we come back, we look at the tubes, and we look to see if this gelatin has essentially melted at room temperature, which would be weird. And if it's liquid, we know that it has been hydrolyzed, it's been broken down. You know, you can take a tube like this one that's been incubated for a week, and you can even cool it down. You can put it into a bucket of ice, and it still won't go back to solid because it can't. The protein has been broken apart in there, and it's now just a soup of amino acids. So that's the gelatinase hydrolysis test. This next picture shows the urease test. So the urease test is typically done on an agar slant. And that's what we're looking at here. We're looking at 2 agar slants side by side. So you would smear your bacterial culture onto the agar and put that into the incubator. And we generally incubate these for 24 to 48 hours, so a little bit longer than usual. And that's because it takes a little time for a microbe to digest urea, if you will. Now, this medium has an indicator dye in it, as you can see by the picture. The indicator dye in here is our old friend phenol red. And if you recall, phenol red turns yellow in acid conditions. At least that's how we've seen it used so far. Now, in the urease test, the medium itself, the agar itself, is slightly acidic. So before we even put bacteria on it, the agar is going to have this yellow color. And that's because the agar itself, with the ingredients for this test, has an acidic pH, a slightly acidic pH. So when you take this tube out of the box, it's going to be yellow. Now you put your bacterium on it, you incubate it for 24 to 48 hours, and then you look for this color. This has to be my favorite color of all of the metabolic tests that we do. I love this color. They refer to this as fuchsia. This is called a fuchsia color in the urease test. And remember, what's happened is the urea has been broken down and it has produced ammonia. And ammonia is a basic chemical. It has a high pH. And in the presence of phenol red, when you have a basic pH, the medium turns fuchsia colored. So a negative result would be a yellow tube, and a positive result would be this gorgeous pink color. So those are the two tests that we use for whether or not a microbe can digest proteins or a protein waste product, urea. Now we'll look at the two tests that are testing whether or not a microbe can catabolize an amino acid. And I'll say we're getting towards the end of our 4 o'clock time. So if I get cut off, just join in again at 5 o'clock and we'll keep going. So the first test we're looking at is the phenylalanine deamination test. And this one is also conducted on an agar slant. So inside that agar slant, there's, a basic nutrient medium, but it's supplemented with this amino acid called phenylalanine. And if you recall, the deamination reaction is going to produce an acid and ammonia. So we spread our bacteria onto the slant. We incubate it for 24 to 48 hours. Again, we're going to give the microbes a little extra time to do the metabolic reaction. And then when it comes out of the incubator, we're going to add ferric chloride. And remember, that's one of those iron-containing compounds. So you drop some ferric chloride into the tube, and ferric chloride has sort of a golden yellow color. So over here on the right-hand side, you can see this you can see the slant, you can see the cream-colored agar on a slant, and then you see this gold-colored liquid here. That's the ferric chloride that has been added. Now in this tube, no color change has occurred other than the color of that ferric chloride. In this tube, you can see the green color. So in this tube, we have a positive result for phenylalanine breakdown, for phenylalanine catabolism. And microbes do that through deamination. So this is what a negative result would look like once you added in the ferric chloride. And this is what a positive result would look like. And then I put these in as well, just so you could see another image of these. Here's a negative result. Again, you can see the liquid that's been added into that slant. That's the ferric chloride. And then over here, you can see a positive result because we have a reaction occurring with the iron. That dark green color is what we're looking for. And the last test on our list is for hydrogen sulfide production. Remember, we grow the bacterium on agar, and then we look to see, we add in another iron containing reagent, and we look for black. We look for a black material. Now, I also have two different pictures on this slide, because you can do the hydrogen sulfide test either in a broth or in agar. It really depends on the microbe that you're trying to grow, which one might be better. So this picture on the left, these are broth tubes. And again, they've had cysteine added to them. That's the amino acid that contains sulfur. So this broth was inoculated, it was put into the incubator, and then when it came out of the incubator, we added this ferrous compound, this iron-containing compound called ferrous sulfate this time. And we look for this positive black color. When you do this test in broth, the result is really striking because you're going to see the black color all the way through the broth. Over here on the right, we're actually looking at agar. These are agar deeps. And if you recall, the way you inoculate an agar deep is with a wire. You put some bacterial cells on the wire, and then you stick the wire down into the agar. So it can be a little bit hard to see the growth. You have to look at the little stab mark and see if it looks cloudy around the stab area. It's tough, it's hard to see. But when you're doing this test, this desulfuration test, remember, we're looking for a black color. And I think you can see that there's black all the way along the stab line where this tube was inoculated. So this would be a negative result for hydrogen sulfide, and this is a positive result. You can see where that wire was stuck down into the agar with the bacterial cells. And you can see that as the cells grew in that little poked area, as they grew, once we added the ferrous sulfate, we produced hydrogen sulfide. That's the black color. So you can do this test in broth or in agar. It depends on the microbe. So if you're trying to test a microbe that you know doesn't really like oxygen, you would probably choose to use a deep instead, because it'll allow it to grow deep down in the agar and stay away from oxygen. All right, the last two slides in this set are telling you exactly what you need to do and not do in your lab manual. And of course, these slides are up on Canvas for you. So in the lab manual for protein catabolism part 1, that's on pages 129 and 130. I've gone through and I've told you exactly what you can cross out and what I want you to complete, okay? And then on this next slide, this is for protein catabolism part 2, which is on page 135 and 136. And again, I've told you what you can cross out and what you should complete, okay? So I haven't given you results today. I haven't shown you any results, but you really don't need them. All you need to know is how to interpret the test and what we're exactly testing for with these tests, okay? So, so I've got that up on Zoom for you, up on Canvas for you. Now, the other thing I want to mention, I sent out a message earlier today, but I want to mention that I have been making recordings of these lab sessions on Zoom, and I am now posting those recordings to our YouTube channel. So if for some reason you can't make a lab, or maybe, you know, you make the